Hello, everyone. I think we can start with our webinar. First of all, I hope you are all doing well. Welcome to our webinar, and thank you for joining us today. We are glad to have you today with us. So we will be presenting you the upscaling of crystallization towards a full continuous crystal production. Bart Rimez, one of Secoya's co-founder and our expert in crystallization, will be the main presenter. Bart will showcase our crystallization technology and demonstrate how this technology can be seamlessly scaled up from laboratory scale to production scale with a same set of parameters that is tightly controlled. For my part, Diala Yusevic, Technical Sales Manager at Secoya, I will mostly have a role of a facilitator. And today we have the chance to have Jan Vogeler from Fette Compacting, who will demonstrate our combined and joint effort and highlight the interest of collaborating together. Just for your information, we have questions and answered a Q&A tab that you can see on your left-hand side on the screen. So all the questions will be written in the tab during the webinar. The question will be addressed after Bart's presentation, and all the questions will be answered formally and shared with you after the webinar by email. For your information, all the questions are anonymous, so please don't hesitate and take care on this aspect. We will also have a poll question during our webinar, so 3 of them, and the poll result will also be shared after the presentation. For your information, this webinar is now being registered, and there will be a link that will be shared with all of you afterwards if you want to share it with some colleagues or look at it again. Secoya Technologies has today 4 technology pillars. So we have developed for these past years equipment, but also services in crystallization, which is today the focus of this webinar. But we have also developed equipment in another technology, which is the emulsification and the encapsulation in order to produce single or double emulsion with a tight control on the size and the distribution of the particles. And we have a third technology in pervaporation, which is a liquid-liquid separation through a dense and selective membrane. We also have a fourth technology pillar, which is a process intensification. For this part, we don't have equipment, but it's more an expertise in order to help you in optimizing your processes. I will let Bart take the lead on this webinar and present our crystallization technology.
Thank you very much, Diala, for your kind introduction. So I'm Bart Rimez, the developer of this crystallization technology. Just as a short introduction to the technology for people who are not completely acquainted with it already. Secoya Technologies focuses really on spontaneous nucleation of a material in solution, whether or not with the addition of an anti-solvent, which is depicted there on the left in the animation. What is really the goal using our capillary reactors is that we want to reach spontaneous nucleation conditions as fast as possible inside of these reactors. And thanks to the very tight control in evolution of temperature of this material, you can then really point and determine the point where the spontaneous nucleation of your material will take place inside. And then the tight control of the residence time and the temperature inside of the material makes that we can always produce a material that has a very tight control of both particle size, but also particle size distribution. And we can do that for both cooling crystallization, but also with adding the anti-solvent in different modes, which I'm going to explain a little bit later. Now, for the cooling crystallization, that's really an ideal system where you want to generate oral dosage forms of your material, targeting what we call larger particle sizes in the range of the 50 to the 100 micrometer size, which tends to work really well for every compound in BCS class 1 and 3. Whereas if you have the anti-solvent modes, you can more easily target smaller particle sizes, but also work with more difficultly soluble materials in vivo, as we have found already. And this graph here really shows you the complete size range that we already have achieved using our technology. So from oral depot, oral dosage forms into nasal applications, always going to for smaller and smaller particles, down to really tiny particles as adjuvants to, for example, vaccine formulations. Those are the things that we already have done and demonstrated on our equipment. Basically, our technology facilitates the use of any material in solution, and that up to 85 degrees C, as we see that as a useful temperature limit, let's say, of many molecules in solution before they start to disintegrate, for example, but also many organic solvents are not useful at temperatures above these 85 degrees C. The anti-solvent side is typically something that also is allowed to be both administered heated or cooled, and that's really important for polymorphic control of the system, whereas the reactor, so that's the point where the crystallization and the nucleation, in fact, takes place, can be changed with a decent temperature range of anything between 0 to 70 degrees C, which we found most useful. These things come with different inserts and reactors, and I will explain you a little bit more in detail why this selection needs to be done, how this selection is done. But most importantly, it is that the moment that you have selected the correct insert and reactor on the laboratory gram-scaled instrument, which with your first parameterization of the system, you then keep that combination of insert reactor with also all these temperatures and flow rates constant up to pilot scale, where you do kilogram testing to test your recipe that you have generated at the lab scale, to finally outscale to the SET ICE instrument, again, with exactly the same insert and reactor combination. I leave this to Diala.
Yes, thank you, Bart. So we have our first poll today. So please don't hesitate to answer. So the question is, when studying crystallization, have you considered specific nucleating condition to control polymorphism and or the particle size? The different answers. So first one is never, because you consider that it's impossible. Second answer, never, as production doesn't allow it. Third answer, occasionally for R&D purposes, or you have already done it with success.
Okay, thank you, Dila. Now let's get into a bit more of the details of the technology and how exactly do you attack crystallization inside of our reactors. This basic example here of brivaracetam is the molecule in which the technology development really started. And one of the benefits of this molecule is that it has a tight competition with the desired polymorphic form, desired morphic form, the orange solubility curve there, and the undesired solvated structure, which is depicted here with the blue solubility curve. Now, what typically happens inside of any kind of vessel of any size is that whenever you cool down the system slowly to a temperature just inside of the supersaturated zone here, you will immediately generate the unwanted solvated structure. And therefore, and that's the main benefit of using these really fast cooling rates that you can achieve in our equipment, is that you can really quench this system cooling down inside of our capillary reactor down to the target nucleation temperature in which you will have as a metastable form then the wanted form, or this nice cubic rectangles that you see there. And those are then the ones that are nucleated at these temperatures. Now, the moment the residence time inside of the reactor then is then used to conduct the complete nucleation of the system, and at the outlet of the system, and in this case, we needed to heat up the system to grow to its equilibrium point, since we needed to pass the enantiotropic point there above 20 degrees Celsius. And so in this case, we are able to have polymorphic control of the particles, and then you also using hydrodynamics, you're also able to steer the particle size in the end. Because that's then one of the main benefits of using Secoya crystallization technology, that is that we regard the phase diagram as a two-dimensional issue that we first solve for any new molecule, but then we are really able to play around with the third axis, which is the hydrodynamic axis, which is really enabling us to further increase and to play around with the overall nucleation rate. Meaning that optimizing this nucleation rate can directly generate the particle size of the product that you want to obtain in this case. Just to give you some clear examples on that for both cooling crystallization and antisolvent crystallization. So we have done in the past some work on glycine in water, where we determined a certain concentration, a certain nucleation temperature, and we kept that constant throughout this complete series of tests. In this series of tests, we kept the thermodynamic conditions exactly the same, so temperature evolution, residence time, and all of that. The only changes that were applied is a change in both flow rate and inner diameter of the reactor, together with the length of the reactor, generating always exactly the same residence time, but then enabling the linear increase in shear rate here from 284 per second up to 12,000 per second for shear rate, and as well a huge evolution in the Reynolds number inside of the reactors. And what you see then at the same thermodynamic conditions, and that's quite surprising, is that your nucleation rate J goes through a maximum here, J max, which is then the normalization factor for the complete curve. And when you further increase the shear rate, you start to decrease the shear rate again, meaning that every molecule, in this case for glycine in water, at this particular shear rate of about 200 in this case, here, you go through a maximum in this point. And what's important to notice is that it is a 5-fold time higher shear rate than you would use a slightly lower shear rate, in fact. So your nucleation rate is hugely impacted by these localized shear rates inside of the tubing. And therefore, that's a major part of our work, is to really determine the optimum shear rate conditions for your molecule to nucleate into. On the other hand, for antisolvent crystallization, for example, in this case, I will discuss here briefly what happens inside of a tea mixer, is that you will inject here from the left your heated solution. From the other end, you will inject your antisolvent at a certain flow rate and therefore a certain flow velocity. And you will generate here inside very tiny domains, vertical eddies, in fact, that determine the way that you produce your mix of both the solution and antisolvent. And now here you will retrieve yourself using the system inside of the micro-mixing domains, which is known in the physical world as a Kolmogorov and Batchelor scale, is that you will have these tiny zones in which your antisolvent is in direct contact with the solvent. So whenever you can decrease the size of these vertical eddies, the smaller those domains will be, and therefore the smaller the higher the amount of nuclei that you will generate as a function of time. Just to give you a plain example, we did again some tests with benzoic acid dissolved in ethanol, in this case 60 milligram per milliliter, and we used water as an antisolvent in the same tea mixer. Again, we used exactly the same thermodynamic conditions for both tests. The only thing that was exchanged here is that we have applied a flow rate of 10 and 10 milliliters per minute for both the solution and the antisolvent side. And you do obtain a rather good crystallization with some, let's say, elongated crystalline rods that you will see there. And then simply by increasing the flow rate 2 times, both for the solution and the antisolvent end, you see that you have a huge impact on particle size, but also on the aspect ratio between the length and the width of these crystals, in fact. And so in this case, just by multiplying the flow rates, you increase by a factor of 2 or basically 3 the nucleation rate itself, But on the other hand, you do have also a clear impact on the size histogram of the molecules, with the 10 and 10 milliliter per minute test shown here in green, and then in blue, the 20 and 20 milliliter impact test per minute impact test. And you will see here that you have a very steep increase of the particle size, the size being between 20 and 30 microns. And also the particle size distribution is much better under control in these conditions. So for all cases that we have studied up to now, we are always able to first look for the ideal thermodynamic condition of your system to start to be nucleated. And then by using the different approaches, so this famous third axe of hydrodynamics, we can play around on the overall nucleation rate to better steer the eventual particle size that you want to obtain in the end. So all of this was already demonstrated on both BCS class 1, 2, and 3 molecules, being able also to steer the particle size of the system. But we also have done salt crystallizations, reactive crystallizations, and even amorphous precipitations down to the nanometer scale. And also, what is the benefit of our system is that on one single device, you can have different administration pathways possible by simply changing some of the parameters inside of the test. Just as a plain example, we published a study on the crystallization of aspirin in acetic acid anhydride, where we have used exactly the same concentrations, the flow rates, and reactor length as well. The only thing that was altered here is the nucleation temperature, which is stepwise decreased with 5 degrees Celsius per step in the nucleation temperature. And then you can see that just by simply changing this temperature, you can select a production of 22 micrometer particle size aspirin, or 70, or even 3 microns, depending on the application itself. So that's a true benefit also of this unit, is that you can, by varying several parameters, you can generate a huge, a vast window, let's say, of different powders that can be generated using our system. And now we're also working on different systems, of course. We're in continuous development of performing co-crystallization of different molecules, but also amorphous precipitation towards nanoemulsions as well, and specifically also some work on spherical agglomeration. And I think that makes us the time for the second poll, Diella.
Perfect. Thank you, Bart. So thank you all for your responses for the first poll. For the second one, beside crystallization of different particle sizes, what would benefit your organization most from a single technological platform? Nanoemulsions, which are amorphous stabilized particles, co-crystallization, spherical agglomeration, nanolipid vesicles, or other types of application you could think of. And if this is the case, please do not hesitate to specify it in the Q&A tab afterwards. And thank you in advance for your replies.
Okay, that means that now we can dive a little bit more into the process development of the system. So firstly, you will start out working with the ACT-Lab instrument, which is the laboratory unit enabling to do batch-wise tests of the gram scale using the set combination of insert and reactor. So for example, if you would look at adipic acid, for example, we selected first a cooling zero insert, meaning that we don't play around with hydrodynamics in this case, and a 3 milliliter reactor in all to start as a good starting point, let's say, to conduct the crystallization. We applied a solution temperature of 60 degrees Celsius with a solution concentration of 60 milligram per milliliter as well, and a flow rate of 30 ml per minute. And what we typically do then is that we stepwise decrease in a series of tests the nucleation temperature. In this case, we selected 2.5 as the increment of doing so. and then you will always measure the average crystal length that you obtain of the product slurry coming out of that after filtration. And you see that starting from 25 degrees Celsius, you have this steep decrease, because this is a logarithmic scale, a steep decrease of the average particle size down to the wanted average of 100 micrometer for these particles. We saw that by nucleating at 15 degrees C that we obtained two fast blockages of the system, whereas this was something that we did not achieve at 17.5. So we did not regard here 15 degrees Celsius as a valid starting point. So once you have done this, and we are, in this case, we were happy with the 100 micrometer average particle size with a narrow distribution, because it's the D10 and D90 values are depicted there. We then study already at the laboratory scale, we study the crystal growth. So what we did for adipic acid is that we gathered slurry coming out of the reactor of a test performed at 20 degrees Celsius, which is indicated here by time 0. And then you let that slurry grow for, in this case, the first one, 15 seconds. You filter that product. What comes out, you analyze, and you depict here the D10, D9, D50, and D90 values. And you see that by increasing the time in between the coming out of the reactor and the filtration, that you have an increase in particle size, which is logic because we saw in the previous graph that at 20 degrees Celsius, we were more in the region of something close to 200 microns. But you see that the sample filtered after 90 seconds or 2 minutes, or even after 24 hours, doesn't really evolve anymore in particle size, meaning that, and that's something that we also have verified by studying the mother liquor, is that after 90 to 120 seconds, you already have obtained the equilibrium phase, meaning that you cannot have more crystals coming out, or crystal growth is arrested. So that's a good point in time for to be used at the pilot scale to already apply the filtration of the unit. So then, keeping this recipe as depicted here, we then translate that to the pilot scale. So in the pilot scale unit, which is a kilogram scaled unit, you will have here inside your vessels, which can be heated and cooled exactly as in the laboratory scale, but you can introduce several liters of product inside. What is not changing versus the laboratory scale is the combination of insert and reactor. The only thing that really changes is that you apply the flow setup of the industrial scaled unit. So you will use exactly the same pumps, the same pressure control, the same flow rate control. It can be executed as a fully automated unit already, because we will use the same software as I will show you later of the ICE unit. And it can be in a GLP or CGMP version as well. So using this unit, we prepared an 8-liter solution of adipic acid so that we would be able to conduct a test of about 4 hours fully testing on the unit with, like I said before, exactly the same recipe, so keeping everything constant. And then you would see here that by using these different times after which we had a sampling of several minutes of product, waited then for 2 minutes. That's why it's also these increments of 2 minutes before we applied the filtration. And then you see that the D50, but also D10 and D90 values do not change as a function of production time, and are or let's say, completely similar to the values that were obtained with the SCT lab unit. The only outlier you might consider here is the D90 after 224 minutes, but which seems to be just more of an outlier, but nothing else. So, in fact, this kind of symmetry that you obtain in these graphs indicate the stability of the product that you would have over time. So we can maintain the same product quality over time, but you can also fully extrapolate all the lab parameters that you have set out before. So you don't lose any time anymore with the upscaling, because the parameters were already set before. Now, the same example can be done for lactose in this case. So now we cannot completely disclose whatever we have done here on the execution of the insert and the reactor. and because of confidentiality reasons. But what we can share is the fact that we have used here a very high concentration, because lactose is a very soluble material inside of water at 85 degrees Celsius, with a flow rate that we applied here of 20 ml per minute, and as was found, an ideal nucleation temperature of 5 degrees C. We have to remark here that lactose is a good nucleator, but a very slow growing material, meaning that we had to put lactose inside of waiting tanks at 5 degrees Celsius for 18 hours in order to completely grow to its equilibrium state of these nice, what they call tomahawk-shaped crystals like you see there. So again, what we did here for lactose, after a complete optimization scheme that I'm not going to show you here because it took us some time, especially to find this execution for highly viscous materials in this case. But these different laboratory results here show you that, again, we do have a stable or an acceptably stable output of material independent of the test that we have performed. And then by keeping, again, this recipe exactly the same, but then performing 2-hour runs of this unit. And I have to remark, these runs are always done without any sort of disruption in the flow rate or any other event occurring. You see that, again, the results obtained after passage inside of the pilot tests are exactly the same as the laboratory tests, even though we have to wait here during 18 hours. And now to check on this, we did perform batch cross tests where we did exactly the same concentration, the same temperatures, but then not passing the material through the reactors. And then you see that you have a steep increase in average particle size. So even though that the passage through the reactor takes nothing but 45 seconds, more or less, you do generate a huge amount of nuclei that are then simply grown to their equilibrium state inside of the tank. You still have, when you don't pass through the reactor, a crystallization events occurring, but you do see directly here that you have these big lumps occurring, that there is a wider distribution, but most importantly, you have a 50% increase in particle size with respect to the pilot tests that we have conducted. So that's an important fact. And then, of course, you're able, when you have conducted these recipes at pilot scale, you also have done your boundary batch tests to see which conditions, at which boundaries, which conditions can be set. You are able to increase your scale to the industrial crystallization equipment, or SCTIs. The ICE unit here is based on, again, an identical reactor setup of your laboratory equipment. And then you simply multiply, in this example here, which is shown in this picture here, we have multiplied the amount of reactors by 5, but with the current demonstration system that's sitting here in our building in Louvain-la-Neuve in Belgium, it can accommodate up to 10 of these reactors in a second table here on top. We have created a molecule specific or a multi-product approach, meaning that you would be able to exchange these modules to be accommodated for a different molecule. But that's really something we see that need to be negotiated together with the client. It has an automated software which has an open communication platform also to its surroundings to be introduced into your networks, so that it has the possibility to fully communicate with systems prior to our unit, but also maybe to any filtration unit that comes behind in the production scheme. Now, in our quality by design approach of our unit, it's really important, since the reactors do not change anymore in between laboratory and industrial scale. It's really important to both have as stable as possible nucleation temperature, which is depicted here on the ice running after the startup of the system. To have a stable temperature which does not vary anymore, which is hugely important. And then we have generated an algorithm that opens automatically the modules whenever all temperatures here are retrieved as being stable, meaning that you would then open reactor 1, 2, and 3 consecutively, And then this depicts here the stability of the flow rates that are achieved inside of these reactors. Now, the stability, of course, is hugely important for these things. And we do have a real-time parameter control to enable fast adjustments of the flow whenever necessary. However, we are able to generate such a stable flow that they only have a 0.3% deviation of the initial value that you have set out. Meaning that whenever you have a boundary condition, in this case, for example, of 19 and 21 milliliter per minute, for example, in which you see that your product quality is starting to go down, well, you know that you will always be inside of this zone here. And that's then also programmed inside of our unit. So let's say that for this case, the parameter that we set for the flow rate needs to be between the boundaries of 19.95 and 20.5. And we do know that you do have, under normal operation, no variations outside of these boundaries. But we did the tests on the pilot scale, and we know that below 19 and above 22 milliliter per minute, you do have a deterioration of your product quality. So it means that whenever there would be some kind of hysteresis and some sort of deviation in the flow rate, which does not go or does not reach this boundary condition, there will only be a soft alarm generated, and your flow regulators will be able to automatically adjust this whenever possible. You'll go back to the normal flow rate, and there will not be a true event of the system because your product quality is not affected. On the other hand, if there would be like any dust particle or an eventual blockage of the unit, you will see a drastic decrease of your overall flow rate happening and occurring rapidly. And that's then the point where the system will decide, whenever I'm too long outside of these boundary values, to shut down that particular module so that your product, your overall product, is not affected. Your production rate will be a little bit lower at the point in time. But as we allow hot swaps of the system, you can exchange and that particular module and then set this up back again. So if you look at the software that you have generated for this equipment, it looks like this. So you have your different set points for the solution end, and also the wire heating to keep everything at temperature right before the entry inside of the modules. But also the anti-solvent temperatures, the different back pressure regulators, and also the pumps for the complete peripheral system with these different hysteresis temperatures that you can program yourself with, and the current stabilization time, but also these deviation times. And you can do the same programmation for every module. Also, you can copy these set points for all modules, but you can also select different modules having different, well, not temperatures, but flow rates. That's something that we can accommodate. And you have the same kind of programmation there. Introducing this unit then into a complete production environment means that you will have here central point your unit. Inside of this ice equipment, we have two different buffer tanks. In the case of cooling crystallization, it's only the solution here that will be present, of course. Prior to that, you will have solution buffer tanks where you can prepare your solutions, put them at temperature, and then administer them to the buffer tanks accordingly. At the outlet, your products are gathered, and then there's always the choice of both letting the slurries grow inside of waiting tanks at a certain temperature to reach equilibrium state, or in the other example of adipic acid, for example, that you only have to wait inside of these filters for 2 minutes before you do the full filtration of this system. So to give you a bit more insight into how we do this, I will show here some anonymized production strategies that we have developed together with customers already, because that's getting more pertinent, of course. In this case, the customer wanted to have a 25 kilogram production per day of a compound which was in BCS class 2. It's also considered as a potent compound. And they wanted to produce 100 kilogram per week of this compound. Now, we could do some tests only during the day, but they have a 2-shift basis in working hours of their operators, meaning that we would have a window more or less per day of 12-hour production, in which we needed to do the setup, the production, and the cleaning in place of the system. And by calculating everything, we came up with a possible solution, taking all solubilities, the yields, and whatever point that we have been discovering on the pilot scale unit with a 30 milliliter per minute that we could achieve as well. And that we have here in the end a 350 gram per hour per reactor of productivity, meaning that to obtain this production rate, we need to apply 8 reactors that can be produced during 9 hours, a total of 25.2 kilograms. So that's okay. But we will still propose to the customer to use it with 10 reactors to reduce this time a little bit more, but also to encounter any possible event, meaning that you would lower instantaneously a little bit your production rate in order to be able to fully conduct this kind of system. Another example, and this is the lactose example. So our customer there wants to produce 50 tons per year, split in weekly productions. So meaning that we have to produce from Monday to Friday, but from Monday to Friday also including the cleaning in place of the system, but also to conduct this very long 18-hour growth time necessary for the last portion of the produced slurries. And therefore we selected 3 growth tanks to be filled during the week, meaning that during this 5-day work week, we apply 3 full days of nucleating inside of our reactor on the conditions that are set there. We can produce almost 800 grams per hour per reactor in this instance. That's possible, of course, thanks to the high solubility of lactose in the system. And then we can, which we consider the limit of our unit, with using 50 reactors, we can produce per 3 days nearly 3 tons of product, meaning that with 17 of these repeated batches per year, that we are able to both accommodate the full production of 50 tons, which gives us a lot of spare time for every event that may occur, but also to open the space whenever the production needs to be increased. And then a third example that might be considered as a bit of a specific example, but because in this case, it's a reactive crystallization of an inorganic salt. And this inorganic salt is very specific because it doesn't require a reactor to be used. In fact, it's only the quality of mixing in this case that determines the product quality, like I shown you before with this example of benzoic acid. And in this case, we're able to increase the overall flow rates up to 200 milliliters per minute for the solution side and to 1000 milliliters per minute for the anti-solvent side, because we need this 1:5 mixing ratio, again, to be polymorph selective in this case as well. With lower quantities of anti-solvent, we do obtain the wrong polymorphic form in this case. And for this customer, we label with 4 of those reactor, in fact, nozzles that are implemented directly inside of inside of the nutshell cell here, that we can inject with these 4 nozzles and obtain a production of 5 kilograms of solid products per hour. This for them is in fact a pilot scale unit, and we will then increase with them multiplying the amount of nozzles that are generated here on the hood. We can then, sorry for that, we can then still increase drastically the production rate per hour, which ends up with the next poll, Diella. Diella, you're in mute.
Sorry for that. Thank you, Bart. So our last poll is, and our last question is, how would you consider the implementation of this technology at your company? Would you be interested to have a full in-house development and production cycle, which means going from the lab equipment to the pilot and the industrial unit at your premises? Or would you go for first feasibility studies done by Secoya in our premises, then implement also the lab, the pilot, and the industrial unit? Or would you go either to a feasibility studies and a full process development, a process development at the lab scale at Secoya, then implement the pilot and the industrial scale? or would you rather go for a process development made by Secoya, which is the lab and the pilot unit that we would do for you, and then implement the industrial scale at your premises?
Now, of course, we can produce these nice powders with good particle size control and particle size distribution, but we also regard our technology as an enabler technology, let's say, to overcome the different prerequisites between what is a drug substance production and the drug product manufacturing. In fact, because if you look at the drug substance synthesis, we can control polymorphism and size, but for the real drug product application, you do also need the control of density, flowability, compactability for both dry or wet granulation. But we truly see our unit being able to bridge the gap between DS and DP, thanks to the fact that we can do control and have a very uniform output of material, having always the same density, flowability, have less issues, for example, with electrostaticity, increasing generally the feederability of the product. And therefore, we're really happy to announce that we will work together with the Vette Compacting Group, and that allows me to introduce Jan Vogeleer, which is the Director Global Product Management and Strategic Business Development of Vette Compacting, to introduce their continuous manufacturing equipment and to discuss briefly how we can introduce our Secoya crystallization technology with their continuous manufacturing equipment. Jan, the floor is yours.
Okay. Hey, do you hear me?
Yeah.
Okay, good. Yeah, Diana and Bart, thank you very much for inviting me to this interesting webinar. Yeah, I was already introduced. My name is Jan Vogeleer. I am based at the Vette Compacting Belgium office, which is an entity we have created back in 2017 in Belgium, in Mechelen, between Brussels and Antwerp. And the Vette Compacting Belgium is not a sales and service subsidiary. We are in fact a development and launch entity within the Vette Compacting Group. Vette Compacting, for those who are not familiar with us, we are a company based just outside Hamburg, and we develop and sell worldwide rotary tablet presses and also capsule fillers since shortly. We can say we are market leader in industrial tablet production based on this concept of rotary tablet presses. Now, some years ago, we decided, in the interest of expanding the activities of the Vette Compacting Group, to step also into continuous manufacturing. We are active in the area of drug product production, where since 15 years, I would say there is a strong interest and a bit of a slower trend towards continuous manufacturing. Our industry is a batch manufacturing environment, and for obvious reasons, we want to go to continuous manufacturing. Now, what the link is with a company like Secoya, I would like to explain in a few minutes. So yeah, if we can go to the second slide, please. I don't know who is, I cannot, yeah, okay, thank you. Yeah, you can also appear the text which is on the right side, please. So why does our industry, the drug product manufacturing industry, why is there this interest to move from batch manufacturing to continuous manufacturing? Well, there are 5 main reasons, which are listed here on the right. The 2 obvious ones are when we interconnect all those different unit operations, as we call it. You have the dispensing, mixing, granulation, tableting, coating, packaging. If you combine all those unit operations into one long chain of subsequent units, we have, of course, some obvious savings like reduced capex. The whole system becomes smaller, more compact, and therefore also reduced cost. You need less space to install it. which is also a big saving in manufacturing area. And of course, you don't have the all the intermediate product stock. This work in progress is significantly reduced. So that's the reduced capex and opex. An important cost-saving element is the fact that there is no need anymore for intermediate quality control between the different process steps. But what is much more important advantage of continuous manufacturing is the point 3, 4, 5. So point 3 is more efficient production. The size of a batch in our industry now is determined by the size of the machine. Now, if you have a batch coater or a batch fluid bed or a batch granulator, the size of the machine determines the batch size. Tablet press is the exception there. Now, when going to continuous manufacturing, the batch size is not determined by physical dimensions of a machine anymore, but by runtime, which of course gives a lot of flexibility in terms of how big batches I can produce. So efficient, just-in-time manufacturing is an important advantage. The other one is that when connecting those different unit operations into one continuous production line with raw materials in on the one hand and the finished tablet on the other hand, is that you can control the entire process and the quality of the product being produced by one central control system. So you can have feed forward and feedback control loops from one operation or one process to the other. We can measure things in the coater, which can allow us to optimize the compression of the tablets. Also the other way around, we can detect certain things in the tablet press, which allow us to adjust parameters in the coating process. At the same time, new sensors are being developed, many of them based on spectroscopy, like near-infrared spectroscopy, are being developed to be integrated in the process to do a permanent monitoring of the quality of the intermediate product and the end product. This, again, to eliminate this laboratory testing. But the last but not least is the fact that continuous manufacturing and having all those unit operations closely connected to each other is that it allows process and formulation developers to develop new products much faster time-wise, using much less material. Basically, you can turn on the knobs of the tablet press or the feeders, the blender, the granulators, and you see within a few minutes you see the impact on the final tablet. And that is for most of the companies the biggest driver to step into new product, into continuous manufacturing, and then especially for new products which are being developed. So that's why the industry wants to go from batch to continuous manufacturing. So next slide, please. Now, as I mentioned before, around 2005, there were the first pharmaceutical companies and equipment suppliers who stepped into the development of continuous manufacturing technology. And the initial focus was on continuous high shear granulation and drying. As a matter of fact, Professor Raymond at the University of Ghent developed back in between 2000 and 2005 the twin screw wet granulation system, which was then adopted by GEA and turned into a commercial product. Now, the thing is that we are now 20 years later, and only about 20 drugs are being produced in a continuous way by less than 5 or more or less 5 companies. So continuous manufacturing is really not taking off very much. The reason for this is, in fact, three points. The equipment, well, there was a big focus on continuous high shear granulation and drying. Yeah, that was the main focus by those early movers. And why? Because in the batch world, about 80% of all drug products are being are based on a wet granulation and drying process. Now, when switching or developing this continuous manufacturing technology, there was big focus on developing a continuous granulation and drying technology. Now, this made equipment very complex and therefore very expensive. It also led to the fact that equipment was made very customized. Every customer had his own view on how to build such a continuous line. And so, in fact, every time a very customized system was built. So that led to a huge complexity, long delivery times, and also uncertainty with regards to how are we going to do the regulatory filing of this new production process. So that has resulted in our vision that when we want to make this switch from batch manufacturing to continuous manufacturing in the industry a success, we need to go away from this granulation-based processing to a simpler process. Now, why are products being granulated prior to compression or prior to capsule filling? Is in fact to achieve three things. That is, in case of tablet production, of course, we want to have good flowability, because if the compression blend does not flow very well, the die filling of the tablet press will be irregular and the weight of the tablets will be irregular. So we want to achieve good flowability. We want to achieve good compressibility, because if the tablet does not compress or the blend does not compress very well, we will have all sorts of problems like capping, lamination, broken tablets in the blister process, etc. And the third thing we want to achieve is we don't want segregation, because we do all those efforts to make a perfect blend, etc. And then we put the product on the tablet press, and while it flows into the tablet press, the product starts segregating. That's not what we want, because then the content uniformity of the tablets is not within specifications. So that's a reason to do this. There was a reason in the past to do wet granulation. Now, as we see it, we strongly believe that due to this complexity and high cost, etc., continuous manufacturing in drug product manufacturing is not advancing as it should be. And we believe strongly that by switching to direct compression, that is the thing that will enable the industry to embrace continuous manufacturing on a broad scale. Now, that's why FETA has developed its own continuous direct compression system, which I will show in a minute. But in order to make continuous direct compression possible, we need an alternative solution to this granulation in order to avoid segregation, have good flowability, and have good compressibility. And we strongly believe that the solution for this is not in what we call the formulation development, but upstream in the production of the drug substance. And that's the exact reason why we, as FETA Compacting, are interested in what Secoya is doing as a technology, because we believe that novel ways of API production will enable the use of continuous direct compression. Sorry, can you switch to the next slide, please? Yeah. So here, basically, you see the solution which we have developed and which we brought to the market at the Interpack exhibition before just a month ago. So you see here on the left side, one of our tablet presses, the FE55. In fact, it is the tablet press which has the highest output on the market for a single-sided tablet press. Single-sided means one product inlet, one tablet outlet. What you see on the right side is the FE-CPS, the continuous processing system. And basically, this is a system which will convert the different raw materials, combine them together, and make a compression blend out of it, which is then fed directly into the tablet press. Next slide, please. Yeah, so here you see the main components of this system. So you see on the top 6 material inlets. That's typically API, a filler, binder. There will also be a disintegrant, and nearly in all cases a lubricant. Now, why do we have 6 material inlets? Well, a study has shown that 85% of all tablet formulations have 6 or less components, ingredients. Now, there are, of course, tablets which have more than 6 ingredients, but in that case, we typically work with what we call a premix. So you have multiple minor ingredients, which we then pre-blend into one bin prior to bringing to the line. So up to 6 material inlets, we have what we call automatic refill systems. That is important to dose how the raw materials are fed into the machine. And below that, you have then 6 loss-in-weight feeders. The loss-in-weight feeder is a very accurate, very precise device that is dosing each of those 6 ingredients at a very accurate feed rate, expressing grams per hour, kilograms per hour, grams per hour into the blender. In the lower section, you see the blender. All in the bottom, you see the blender. Thank you for indicating. And between the loss-in-weight feeders and the blender, you have what we call the funnel configuration. In fact, that is an exchangeable system which determines which of the different ingredients is entering the blender at what point. The blender, without going into details, is a system which Feta has developed, designed themselves. It is a blender which has different blending zones with different blending intensity. Some materials need high intensity mixing. Other particles need a more gentle mixing to avoid, for example, over lubrication in case of magnesium stearate. So we have different intensity zones, but also different inlets. Yeah. So the funnel configuration determines which of the 6 ingredients is entering the blender at what point. At the outlet of the blender, where the arrow is now, we have a possibility to measure the blend uniformity. I'll come back to that in a second. And from there, the blend is then conveyed and lifted about 2 meters high, and then transported into the inlet of the tablet press. You might now say, whoa, you do a lot of effort to make this perfect blend, and then you just convey this blend into the over a distance of in total about 4 meters. Well, yes, we do so, but we do that with a very special dense phase blend conveyor. It's a technology which is available in the market, but which we have to fine-tune to make sure that we convey a compression blend without any risk for segregation into the tablet press. The fact of working like this results in a system which you see here, with a total height of less than 3 meters. And that is what allows this system to be installed in an existing tablet production facility. So it is a very compact system which fits in an existing production environment. A last important point to mention is I said in the beginning of the presentation that one of the big advantages of continuous manufacturing is that the total production, the total production process is monitored by one single control system. That is the case here, but in order to monitor in detail the quality of the intermediate product and the end product, we have developed this, what we call embedded PAT. So these are various types of spectroscopic-based sensors which monitor the quality of the blend at intermediate stages, but also the quality of every single tablet leaving the tablet press. So,
Jan, if I may interrupt, because I think we're almost running a little bit out of time. Yeah. We will also send everybody, sorry to interrupt you, Jan, but we will send to everybody a link to this nice animation on how this beautiful equipment runs. But what is very important here is also to say that we are looking for actual case studies where we have linked to the industry. So really the provision of powder that can be both treated with our crystallization technology and can be directly measured with all the different parameters necessary to be introduced inside of the FETA compact, the continuous manufacturing compacting machine. We're running out of time, but I just have to tell you one more thing that we have been developing. Because there was big questions regarding the total amount of product that can be used during the laboratory screening phase. And that's why we have developed this unit, the InstaDrop unit, which enables you to do the full screening of your material with regarding polymorphism, but also interaction with different solvents by encapsulating really small dosages, small amounts of material inside of a polymethacrylate resin that is then hardened out using UV light, upon which you can do then later on all sorts of exchanges with other liquids to be able to generate, for example, like in this case, single crystals inside of encapsulated materials. And so we did already a huge range of different materials that were crystallized on copper sulfate, sodium chloride, but also proteins and different organic system. But the main advantage here is that per test, and you will do several thousands of capsules that you can produce per minute, but with only using 50 microgram of your very precious solute material in the early stage of the system. And which is really good is that once you have determined the complete temperature, solvents, anti-solvents, it wins you a lot of time before you need to go to the acetate lab environment, especially because the InstaDrop instruments also is accommodated with different ways of microfluidic mixing so that you can scale this readily up also for the acetate lab environment. So winning a lot of time. So really, again, downscaling to the early stage and then doing the complete process development and upscaling the devices using the lab, pilot, and ice environment. With this, we would like to end this session. We're already running late, but just quickly, for the different collaboration points that we do organize live demonstrations on site, we do ourselves, as Diala has already mentioned, proof of concept and feasibility studies for you, upon which the project is then later on handed over to your sites and to your industries. Or we do also do leasing of all equipment with an intense tech transfer included and follow-up during the leasing period so that you optimize your time and reduce the R&D cost and R&D time as well. There is an upcoming event in October where we invite you personally to our premises where we will demonstrate the new version of both the SCT Lab and SCT Pilot units. And we also will showcase the industrial crystallization equipment. And we also will put different application notes and case studies will be made available also later on. Diala, I don't know if there is still some time for a question to take on, because I know that we're running late already.
We're running a bit out of time, but please don't hesitate to put and to share some question in the Q&A tab. We would be more than happy to answer you. We can also share, of course, as I said it in the beginning, the responses with you by email. I see that we have already one question. So thank you for the question. First one is, how would you proceed with the new molecule? Or can you answer?
For any new molecule that we take on, well, the first selections that we need to do is that you need to do a solvent selection depending on the solubility of the material, and also in many cases the anti-solvent selection. But to do that, we also need to know the requested particle size and particle size distribution, let's say, of that molecule. So that means as well the way that you want to administer your crystalline material afterwards. Once that is done, we will step up with the laboratory equipment. I'm just going here a little bit faster. Then the tests are done exactly in the way that you show here for adipic acid. So several concentrations will be made. We will lower the nucleation temperature or vary any kind of parameter and just to enable the optimum thermodynamic condition. And then we'd also do that with anti-solvent additions, testing different flow rates, different mixers. And once that is done, then we will look into the hydrodynamics of the system whenever the required particle size and distribution is not met yet. Then we will optimize hydrodynamics to reach exactly at equilibrium state the particle size that you end up with. And that's then the determination of the recipe, which is then later on translated to the pilot unit.
Thank you,
Maybe
Bart.
one more question, Diala.
Yes, one more question. How to implement your technology for the regulatory bodies? This is a very interesting question that has been asked several times. Bart, I'll let you answer it.
Yes, indeed. Now, we're already in discussion with many partners in the pharmaceutical industry. And of course, we cannot go directly without any readily available material to the FDA and say to them, well, this is our equipment, just have a look at it and tell us that it is good or not. So what we did try to do is to generate a system, and that's by working only with people who are very experienced in the field, who can program for us GMP regulated softwares, to use only the right kind of materials, the hardware, the software. So everything regarding that is already, let's say, at level. But we do help customers with setting up the dossier for the clinical phases that you need to go through. Whereas, for example, clinical phase 1 and sometimes phase 2, you can solve a lot with producing small batches on the SCT lab equipment. The moment that you need to increase your total amount of product, of course, then you need to scale up the system to the pilot unit. And then with the pilot unit, we do help you, of course, with setting up the complete dossier, with determination of the boundary conditions of all several parameters to just showcase the robustness of the production that we are organizing, to describe completely the process, how it should be done, to enable you then later on, without changing anymore the dossier, by multiplication, the amount of reactors that you produce. So we do help a lot of pharmaceutical companies already with this transition. So we know that at some point we will be able to install and do a full commercially production with the customer in due time.
Perfect. I think we can just have, there are two other questions. We're going to go very quickly on them. It's concerning the continuous isolation and drying to combine with the crystallizer, and also a question concerning the integration of a PAT into our technology.
Yes, so PAT can be introduced when, for example, we use a waiting tank and you need to let grow your crystals to a certain equilibrium temperature. There we will be using the size probes from Mettler that are existing, of course, and there you can monitor and control the evolution of particle size and then go for filtration afterwards. Product that comes readily out of the reactor, it goes so fast and it's so tiny, of course, that directly implementing PAT on our reactors, that's impossible. So whenever there is and then I can directly respond also with the continuous filtration unit. We work together with different partners in the field who themselves have PAT included inside of their continuous filtration units. So whenever the product slurry coming out of our reactor, and it does take some seconds of in time to reach equilibrium stage, which is mostly the case for the lowly soluble materials, then the direct isolation and size analysis of that can be done directly and can be coupled back if necessary. But usually, because we have this quality by design approach, is at the moment that we don't see any deviations inside of the flow rates and temperature of the unit, we know that the product quality is guaranteed. So for the moment, we did not implement yet any feedback loops coming from the particle size analysis back to our reactors, in all honesty.
Thank you. I see that we still have some questions. Can we still answer them, or should we just answer them by email? So last question, have you achieved the 90 smaller than 10 micron for small organic molecules.
Yes, indeed. So that's a compound which I cannot disclose, but it was a low soluble compound, and we achieved D90 of 3 microns in that case. And there is also a customer now working up a process, and there the effective goal of the project there was to achieve D90s below the 10 micrometer mark, and they achieved that within, let's say, 10 to 15 tests on the SCT lab equipment even. So it usually depends on the kinetics of the system, how well it nucleates and how fast it can nucleate, but also how it interacts with the antisolvent. But it is achievable, yes, absolutely.
Great. For any further question, please do not hesitate to reach out to us. And we would be more than happy to answer you, to organize some technical calls if needed, and as Bart said, to organize demos on live demos, proof of concept or feasibilities, etc. Thank you all for participating in our webinar. Thank you, Bart and Jan, for your interesting talks, and hope to hear from you soon.
And thank you all for your time. Cheers. Bye.
Thank you. Bye-bye.